of heart. We will be talking today about a Cameroonian family in which we have identified a variant in CLIC5 gene as being the possible etiology of the disease. This study is based on my PhD project at the University of Cape Town in South Africa. Hearing impairment is an inability to hear properly on one or both ears, and it is actually a public health issue. According to the World Health Organization estimates, about 430 million people live with disabling hearing impairment all over the world, which represent about 5% of the global population. And interestingly, 80% of these people live in low and middle income countries. We tried to assess the burden of hearing impairment in the Cameroonian population through a systematic review of the literature, and we found that hearing impairment is also a public health issue in Cameroon with a prevalence in the general population of up to 3.6%, with a more higher burden in the elderly population where the prevalence can be up to 15%. Depending on the part of the hearing system that is affected, we can have three different types of hearing impairment. We will talk of conductive hearing impairment when there is a defect or an injury on the outer audio, which basically means that the sounds are not transmitted properly from the outside environment to the inner ear. We will talk of sensory neural hearing impairment when the inner ear is not able to process the information that it receives. When we have both cases at the same time, we will talk of a mixed hearing impairment. Based on the degree of the loss of hearing impairment can be classified in four different categories. We can have a mild, moderate, severe, or profound hearing impairment. From the moderate hearing impairment, which means that the hearing threshold of the patient is more than 40 decibel, it's most of the time not possible for the patient to have a verbal conversation without a specific clinical intervention such as the hearing aid or cochlear implantation. Hearing impairment can also be classified based on the etiology. So we have hearing impairment cases of environmental etiology and hearing impairment cases that are inherited, which is basically monogenic hearing impairment. But it's true that sometimes we can't conclude on the etiology of hearing impairment, which means that we can't say confidently whether the etiology is environmental or genetics. And in this specific case, we classified it as being of unknown origin. The two previous studies that assessed the etiology of hearing impairment in the current population found that environmental factors are the leading etiology with a prevalence ranging from 52 to 62%, with a low contribution of genetic factors from 1 to 15%. But we think that the contribution of genetics to hearing impairment in the Cameroon population might be underestimated because we can see that up to 37% of cases are classified as being of unknown origin. And in most cases of sporadic hearing impairment cases that did not benefit from a molecular diagnosis, we will classify it as being of unknown origin most of the time. We have two different clinical types of monogenic hearing impairment. We have the non-syndromic form and the syndromic form. Syndromic hearing impairment simply means that in addition to hearing impairment, we have other clinical signs. And today we have a lot of syndromes that have described as having hearing impairment as one of their clinical signs. We, for example, have the Waardenburg syndrome, in which in addition to hearing impairment, we have some pigmentary abnormalities. We can have the Usher syndrome, which associates hearing impairment, and some visual impairment. Non-syndromic hearing impairment simply means that there is no other clinical sign associated with hearing impairment, and it is actually a highly heterogeneous trait with more than 120 genes that has been described so far as being associated with the condition. Connecting genes which encode for gap junction protein are the leading etiology of non-syndromic hearing impairment in the European and the Asian populations. And the most common gene is the GDB2 gene, which is implicated in more than 50% of cases in Europe and Asia. We previously tried to assess the implication replication of GJB2 gene in the hearing impairment etiology in Cameroon population. And to do that, we recruited 46 multiplex families, of which 42 were non-syndromic hearing impaired. We used Sanger sequencing to analyze the coding region of the GJB2 gene, and no pathogenic variant or likely pathogenic variant were found in any of the 42 families. Similar findings were actually reported in other sub-Saharan African countries, including Nigeria, Kenya, and Uganda, where no pathogenic variant in GJB2 gene were described, which means that GJB2, which is the major contribution to hearing impairment in Europeans and Asians, is actually not frequent in the African population. So now the question is, which are the genes and the variants that are associated with non-syndromic hearing impairment in sub-Saharan Africa? And given the fact that non-syndromic hearing impairment is highly heterogeneous with more than 120 genes, as I said earlier, the best way to investigate the etiology of hearing impairment in African populations will be to use a method that can assess several genes at the same time, like targeted exome sequencing, whole exome sequencing, or even whole genome sequencing. So we try to identify the genetic etiology of hearing impairment in a Cameroonian family that tested negative for variants in GJB2, and to do that we use the whole exome sequencing analysis method. Before the beginning of our study, we obtained formal approval from 3 different ethics committees, including one in Cameroon, one in South Africa, and another one in the US. A written informed consent was obtained from all our participants, as we can see on these pictures. Most of our participants were identified through community engagement activities in Cameroon, and for all of them, a clinical interview and physical examination were performed, including an audiometry testing, as we can see, for example, on this picture. We also took whole blood from all our participants, and the genomic DNA was extracted in the Division of Human Genetics of the University of Cape Town in South Africa. We also recruited additional controls individuals that were apparently healthy and without any history of hearing impairment. DNA samples from 5 individuals of our target family were sent for whole exome sequencing, which included 2 affected individuals, their parents, and 1 unaffected sibling. Library for the sequencing were prepared using an Illumina capture kit, and the sequencing itself were performed on an Illumina platform as well. Rates were aligned to the 1819 reference genome sequence, and after sorting and duplicate rate marking, variants were called, they were annotated using ANOVA, and they were filtered later on. And of course, at each of these steps, a quality control were applied to make sure that we only keep high quality base and variant. Several filtering criteria were used to select our candidate variants. We first prioritized variant that segregated with the hearing impairment phenotype within the family, following the most likely mode of inheritance, and we were considering both autosomal recessive and dominant mothers. They were also filtered based on the minor allele frequency in normal database. We selected variants with a predicted effect on the protein sequence, which included nonsense variant, missense variant, stat loss, friendship, and so on. We also retained variants that occur in genes that were previously described as being associated with hearing impairment, either in human or in any animal model. Several bioinformatics tools were used to predict the deleterious effects of the variants and to predict whether they affect the conserved region of the genome. To assess the evolutionary conservation of amino acid residues that were affected by our coding variants, we use a multiple sequence alignment of human proteins with protein of other species. Secondary structure analysis and 3-dimensional protein modeling were used to predict the effect of our coding variant on the structure of the protein. Sanger sequencing were used to validate the variant that were identified through whole exome sequencing analysis, and also to confirm the segregation within the phenotype. We also assessed the prevalence of our candidate variants in controls individual and in isolated cases of hearing impairment that We were able to recruit all the 7 members of our targeted family. All the 3 affected individuals presented with bilateral, progressive, and sensory neural hearing impairment. Their audiogram was similar to this one, so we can see that the hearing threshold is higher than 19 decibel on all the frequencies, which means that they had at least a profound hearing impairment. In addition to hearing impairment, no other clinical signs were observed in our participants, which means that they had a non-syndromic hearing impairment. And when analyzing the family tree, the most likely mode of inheritance was the autosomal recessive one. The average coverage of our target region was about 225, and more than 96% of the target region were covered by at least 10 reads. When applying our various filtering criteria that we described earlier, we quickly identified 2 variants, a missense and a splicing variant in a non-hearing impairment gene, the CLIC5 gene. Using Sanger sequencing, we were able to validate the presence of the mutation in all our participants, and we also confirmed the segregation of the variant with the hearing impairment phenotype within the family. We can see that all the 3 affected individuals are compound heterozygotes for the missense and the splicing variant, and all the other unaffected individuals are heterozygotes for one of the variants. We can see that the missense variant has been inherited from the father, while the splicing variant has been inherited from the mother. Our 2 variants were actually novel. They were not found in any of the publicly available databases that we used, including NOMAD, UK10K, TopMed, or even the DBC. They were predicted deleterious and to affect a conserved region of the genome by various bioinformatics tools. They classified pathogenic when we were using the criteria of the American College of Medical Genetics, and they were not found in any of the 122 controls or the 118 sporadic cases of hearing impairment that were recruited in Cameroon as well. The splicing variant was predicted by the human splice finder to break a consensus donor splice site, which is likely to affect the splicing of the pre-messenger RNA through various possible mechanisms, including exon skipping or even activation of cryptic splice site. The missense variants affect, as we can see on this multiple sequence alignment, a highly conserved amino acid residue. And on the 3-dimensional protein modeling, we saw that there is a shortening of this beta strand on the mutant protein as compared to the wild type, and similar changes were also observed on the secondary structure analysis. CLIC5 gene actually encodes a channel protein which is implicated in the transport of the chloride ion. It's expressed in the inner ear of human. It has been shown to be essential for inner ear homeostasis and the hearing process in the mouse. Deletion of CLIC5 in the mouse leads to hearing impairment. And prior to our study, only one report from China suggests an association between CLIC5 and hearing impairment, which means that our study is only the 2nd report worldwide and the very first one, to the best of our knowledge, to show that association in an African population. When comparing the previous family that was described in Turkey with our actual family, you can see that there are a lot of similarities. Hearing impairment in both families was prelingual, progressive, bilateral, and sensory neural, and the most likely mode of inheritance was autosomal recessive in both families. However, in addition to hearing impairment, they described in their families some vestibular and renal abnormalities, which was not observed in any of our participants. Based on our findings and the study that was performed previously in Turkey, we can say that CLIC5 is a good candidate for hearing impairment, and it should be included in the diagnostic gene panels that we currently have for hearing impairment. It will be important to use functional assay to assess the real impact of our candidate variant on CLIC5 gene and function, which we are currently doing now, but we are still analyzing the data. It will also be important to assess the implication of CLIC5 in other populations all over the world in order to refine the phenotype of CLIC5 associated hearing impairment. There is a need to extend the use of whole exome sequencing to additional Cameroonian families in order to solve more cases. It is also critical to continue with the effort that is being done by the African Society of Human Genetics and the History Africa in building capacities on the African continent to generate and analyze next generation sequencing data. Our work has been possible thanks to the input from a lot of people, and we would like to acknowledge, first of all, the financial support from all our funders, including the National Institute of Health, the African Academy of Science, the Wellcome Trust, the History Africa, the University of Cape Town, Columbia University, and the History and the Hygiene Africa project, which is the project under which our study was actually performed. Thank you, of course, to Professor Edmond Wonkam for supervising the whole project. Dr. Carmen is actually the project manager for Hygiene Africa at the University of Cape Town. Unfortunately, I couldn't have a clear picture of her, sorry for that. Dr. Shaheen Mullah is helping us with the functional analysis. Thank you to Professors Leah, Chimusa, and Sherwin for their help during the bioinformatic analysis step. Many other people contributed to this research project, especially our collaborators at the University of Cape Town, the Columbia University in the US, the Aju University in South Korea, the University of Adelaide in Australia, the University of Yaoundé in Cameroon. Thank you as well to our field workers. They have been very helpful during the recruitment process. Thank you, of course, to our patients and families for participating in this research project. The identification of our target population and the recruitment would not have been possible without the help from associations and schools for the deaf. And thank you to the African Society of Human Genetics for